And then they can listen to us be confused about how to flip the screen.
All right, so now we're on. Now we're on.
Okay. Oh, we're on live? Or did you just hit like live or something?
Yes.
Yes.
Okay.
Still no. Mhm. Do we have to invite people to this?
I have no idea.
Stacy,
Just talking.
do we need to invite people, or do they have whatever they need?
I think people should just be able to come in. Black screen that says live.
Yeah, it says live,
Mhm.
and then we were just over something that said invite people.
Oh yeah.
I think like everybody should be able to see it. Oh, here, can you call Adam? He's on the other end trying to.
Okay, what do you want me to tell him?
Just
Did
saying.
Can you see it? Is it working?
you already dial?
No, it's just a little phone button.
Yep.
Hey. All right. How are you?
That's all I got there.
No worries. Have you used the job? Almost. I sort of started looking at one of them. All right.
Nope, you're off by one. This is actually Josh. Hey, I'm just troubleshooting the what are we doing here? Google Hangout.
Oh yeah.
Okay, well, let's see here. We just went up and did the live broadcast. So we have video here. And you're saying you don't see anything? You just have a black screen?
What's not?
Oh.
So, we.
Oh, well, that's good to know.
You're trying to, on Skype, I'm not.
On Google.
On Google Hangouts. And the problem is, it flips the screen.
Yeah.
Who's trying to do
These
that? So
guys.
is one of your advisors, is this trying to go to one of your advisors?
It's actually, yes and no. We have another way to Skype in my advisor. But my advisor is going to, or one of my group members wants to use the live stream, but we have another way to Skype in if the live stream doesn't work. Most of the live streams for my collaborators. Always.
And what do you think
You got it?
So
Okay, great. So
Oh,
you
good.
see the thumbs up?
So Katie and Josh.
Got the thumbs up here?
It's in place because the cable is like weird.
How are you?
Yeah.
Oh, good. How are you?
Okay, well, this one's me, and then that's
all right.
all right.
So on Google, and so I see it. So it flips it, doesn't it?
Yeah.
Hear it?
We're not sure how to get it.
So try it. Yeah, so
Under
we're using a
image.
laptop that's sitting on a tablet that's just using the camera off the laptop. And when it
Great,
looks at the,
wonderful.
it's really weird. Wow, it's a mirror image of the screen.
Okay, all right.
Yeah. Well, in the
Yeah, that's a solid
the-
30, 45
the
seconds.
display.
Good, good.
Showing on the laptop screen, right? Sitting there, hey, Carl, look at the lettering that it's picking up on this projection screen. It's a mirror
Oh, yeah,
image.
There he is.
It's totally reversed.
Okay,
So
sounds
this is
good.
Google. This is Google.
All right.
It's called Google Hangouts or Google
Oh,
Events.
there's Katie, another grad student. She's the one over in the corner. And then Stacy just stepped into the middle of the frame, and Willie is right beside her.
Hang on. This might not. Yeah, we have a different. We have a separate Skype. Yeah, we have a separate Skype. We're all set things. Yeah.
Oh,
yeah,
oh,
we're
he's
on
the
Skype.
building manager.
Yeah, actually, can you, Josh, can you do me a favor and ask Adam if he's okay with slides being backwards?
Oh,
I think we got
hey,
it.
Adam, we just got one question from Stacy. Are you okay with the slides being backwards?
We're not sure how
They're
to
not
fix
backwards?
it.
Oh, it's
Oh, they're not
okay, so
They're
backwards?
not
Stacy-
backwards. Okay,
this is
on
probably
their end,
your
it's not
view.
backwards.
On their end, it's not
Okay,
backwards.
good. Thanks for
Wow,
asking
that's even
that.
better.
Yeah,
Wow, that's even a.
I was really
All right,
worried
so
about
we're-
that. Oh,
Okay.
ask him if
Well,
the mic's working.
Yeah, he's at the audience.
Good.
All right, in that case, I'll hang up and let you enjoy the show. All right, take it easy.
Awesome.
That's what we were saying. was trying to get in. We can actually get into the settings of this camel chart.
Yeah.
Yeah, but if they're okay at that end, we won't touch it. Will, you all set? Everything good?
Thank you so much. This is kind of fun.
Yeah.
Okay, John.
Yeah, look at this. This is my life. Or actually, what's Adam's name? Adam's I mean, I think we'll be okay with the live stream. Okay.
Yeah, he just said that there's 34 times.
Josh, can you just get Adam's name and I'll just add Adam right now so we can just avoid the call later.
Sure, I don't have this.
So, Josh, I have Adam's number.
No, I don't. I'll get that from you.
Yeah. Yeah. Yeah, I'm really excited.
By the way, I found the other one as well. Well, we're going to go ahead and get started here. I can have everybody sort of sit down. It is an incredible privilege today to be introducing Stacy Farina for her defense talk. It's been so much fun and a very rapid period since she arrived here. I think anybody who knows Stacy knows about her independence, the ability to work completely on her own if necessary. She's funny, talented, determined, and her students are now calling her Dr. Branchiosity, which I think is entirely appropriate. You'll see that after you see some of the material in her talk today. One of the things that was most impressive to us about Stacy when she first came here was her undergraduate background from UNH and the fact that she was one of the first cohorts of the Hollings Scholarship Program that NOAA started to sponsor undergraduate science in marine studies. And I didn't really know anything about the Hollings program until I met Stacy, and it was a great thing. It was 2 years of support. You get to go around the country meeting NOAA scientists and working in labs. It's a really wonderful program. But that wasn't the only sort of award honor that Stacy received during her time as a student. While she was here, she got a Rainey Award from ASIH, which is wonderful because Rainey was, of course, a Cornell faculty member. Had an NSF DDIG, a very competitive program for doctoral dissertation improvement grants. And I'm really excited for her because she has an NSF postdoctoral research fellowship in biology, which is starting in October at Harvard. We're very pleased that she's going to work with George Lauder there. While she was here, she served as a TA for vertebrates many times, I think 3. Evolution, she was head TA for evolution, which is an incredibly responsible job because there were 300 plus students in that class. She did writing in the majors for evolution under the Knight Institute's freshman writing seminar program. And she taught, that course was on exploring form and function of vertebrates. So it was basically functional morphology, which is really a wonderful strength that Stacy brought to our program as somebody committed to the study of anatomy and function. One of the things that really is impressive about Stacy is her ability to work effectively at marine laboratories and to take advantage of these really often very strange environments like SNL, where basically you're working with paper clips and string to make stuff work. And she conducted very effective research at SNL. You'll hear a little bit about some of that today. But she was also one of our most sought after TAs during the time I was director out there. She TA'd for us in anatomy and function of marine vertebrates. I think we did that 4 times. That's really, really great. That was, of course, we developed with Frank Fish, and Frank is still running that course. And it's a very popular one, in part because of the reputation it developed during the time that Stacy was the TA for that class. She also taught ecology of the marine environment out there, which was our answer at Shoals to the on-campus ecology course. And that was a ton of fun. We had a lot of good times with that. And then Farina Harbor Labs in Washington State, which is another one of her most important research sites. She taught functional morphology and ecology of marine fishes. And I just have to say, she has this inordinate fondness for kissing fish. Where she is out here at Shoals in 2007 with a cod, and over there at Friday Harbor with the founder. Pretty interesting specialization there, enjoying those fish. The Friday Harbor crew has been especially important for Stacy. This is the invertebrate ball. You participated in this 3 times?
Yep.
Won it every time?
Yeah.
That's pretty cool. This is the one from this summer, the Salpies, all dressed up as Salps. Kind of fun. I would also point out that Stacy's been a really much-loved mentor of lots of undergraduate research students. And Sigal in particular will go, who finished up a little over a year ago, has a paper with Stacy in experimental biology. Noah Bressman, who's currently at Friday Harbor, along with Captain Korn, who's also out there. They're working on pieces that are certainly going to be published in the next year or so. Stacy's done a lot of outreach activities here. She's involved in EYH, which is a program that brings young middle school science girls to Cornell to see what they're going to do in the way of research, possibly. And the physics of fish, which I thought was a particularly interesting program for high school students here. She was involved in the grad symposium here in the EB several times. And one of the things I think is most important about Stacy is that she's really had an active presence at national and international meetings. She's been more than a dozen in the last 5 years, which is really a great testament not only to her research and her willingness to stand up and talk about it, but also to her ability to network and make friends with virtually everybody. Some of you probably know that Stacy's a fencer, which is kind of interesting because Josh is a fencer too. So I had 2 fencing people in my lab, which is kind of fun. And she's, of course, a singer, and her band, Ironwood, is going to be performing tonight. So we're making a little pitch for people to go and see Stacy sing tonight. I think that's kind of interesting. You defend your thesis in the morning, and go sing in the evening. This is over in Trumansburg. Yeah. And
Silver Line Tavern.
Yeah, we can talk about the venue perhaps, that'll be fun. One of the things that she did that I thought was really crazy was she got involved in a thing called the Festival of Ad Hoc Hypotheses, which is kind of a science comedy effort, right? At any rate, that was a big deal because it's a national thing, and it was 2013, and you're going back next year, which is really cool. Yeah.
I just submitted. I'm not.
You're not definite? I'm sure we can get you back. They love it. And one of the things, of course, that I think is really wonderful about Stacy is that she makes friends with absolutely everybody. So it's no surprise that she has this incredible presence on social media. It's been really interesting to follow her Facebook posts this summer as she's winding up her time with us here and thinking carefully about what it's meant to be a student and what kind of world she wants science to be going on in the future. Also, I think it's really interesting that she was by far, I'm a little dubious about scientists on Twitter. I don't know why. I shouldn't be. It actually is really a cool thing. But Stacy has nearly 300 Twitter followers, which I think is pretty cool for somebody at your stage in a career. And without any further ado, I'm going to turn this over to her. Stacy's going to talk to you about the evolution and functional morphology of gill ventilatory systems of ray fin fishes. This is really an exciting compilation of 4 independent pieces of research that actually come together in quite a nice, cohesive way. And I'm going to let her tell you all about it. Stacy.
Thanks, Willie. Thanks, Willie, for that introduction. Silver Line Tap Room, tonight at 8. This system is a really cool and surprisingly poorly studied system. So what I study is, gill ventilation. So the process that fish use to pump water over their gills. this video is. This is like a written challenge for my computer to play 4 videos at the same time. I think we're in pretty good shape. So fish move water over their gills by basically going through this process of taking water in through the mouth and pushing it out the gill openings. So they start by expanding their mouth, then they expand their gill chamber. That draws the water into the mouth and over the gill openings. Then they compress their mouth and compress their gill chamber. You can sort of get a sense of that, and that pushes the rest of the water out the gill openings. And this is really important for maintaining flow in one direction over the gills. And so pretty much all fishes that breathe underwater do this process, but they all have slightly different morphologies, and some of them have really, really different morphologies. So you can hopefully just get a sense here that they're all pretty much doing the same thing. So they're expand, expand, compress, compress, but they all have these really different anatomical systems. So this is a sculpin, a moray eel, a toadfish, and an ocean sunfish. And the wonderful thing about my system is that I get to go all over, and whenever I go to an aquarium, I get to take these lovely videos of my fish doing exactly what I study, because every fish has to breathe. And so if you study feeding, you don't get these opportunities to go and watch all of your fish do all of these cool things. So I really like the videos. But we know a lot about how fish actually do this pumping mechanism, so I've talked a little bit about it, but what do we actually know in terms of the history of studying the system? So we know that we can model this system as a set of 2 chambers, and of course it's actually, if you kind of think about the head of the fish, it's technically 3 chambers, so you've got the mouth, and then you've got 1 gill chamber on each side. But you can simplify that by just thinking about it as 2 chambers, the mouth chamber, or I'm going to call it the buccal cavity for the rest of the talk. That's the mouth. And then the opercular cavity, or the gill chamber, that's where the gills hang out, and I'll talk about what the operculum is in a second. And then the gills are in between, so you can think about water moving in through this oral chamber and out through the opercular valve, or the gill opening. And each chamber has an opening to the outside, and it also has a pump. So these pumps can be really complex, but you can model them as a simple piston that moves water. And I'll show you what they do with these pistons. They move them not necessarily up and down, because these are fish in 3D space. The morphology is really complex, but basically they expand their chambers, and when they're taking water in, like I was saying, they expand their mouth, expand their gill chamber, drawing negative pressure, and then they switch over to generating positive pressure to force the rest of the water out. So they're doing really cool things. So when you're talking about something like suction feeding, you're just expanding the mouth really quickly to draw water in, and you're mostly dealing with negative pressure. This is one of the only systems where you're actually sort of alternating between 2 different pressure regimes, which makes it really complicated but also really cool. And what that does is it sets up a system of countercurrent exchange. So you have water flowing into the mouth, out the gill opening, and therefore it is always, there are a few instances where it might be reversing, but it's generally speaking almost always going in the same direction over the gills throughout the entire ventilatory cycle. And what that does is the fish have set up their blood flow to flow in the opposite direction of that water coming in over the gills. And so you get this really nice system where the water is coming in full of oxygen, and these are just some numbers for oxygen concentrations in water. And so the high oxygen concentration water is always going to meet blood that needs a little bit more oxygen. And so there's going to be exchange throughout the entire system, rather than having, if these 2 hours are facing the same direction, you would reach equilibrium at 50% pretty quickly. So this system is actually the most efficient way to set up a gas diffusion system in any sort of vertebrate. But these guys have to be really efficient because water actually doesn't carry a whole lot of oxygen. And I just wanted to throw in a really pretty picture of these gills. I don't talk a lot about gills, I talk more about the pumping mechanism, but these structures are super important. And so we're looking into the gill chamber right now. The mouth is sort of facing that way. And water would come out through these, you can kind of see that the gills come together and make a sieve. So these are gills, and they're sort of coming together, and water is coming out through these little crevices here, and that's what forms that really nice unidirectional system. But they actually have to do a lot of pumping to make that happen. So I started out my dissertation thinking about pumping mechanisms in fishes, and one of the first things that sort of grabbed me was, I'm always sort of grabbed by the really weird things. I think you'll notice that as a theme throughout my talk. I like really weird fish. And I noticed that a lot of the weirdest fish have this morphology of having a tiny, tiny gill opening. And I hope now, maybe some of you might have seen that when you were watching the videos initially. These are 2 videos that I've already shown. And there's the gill opening on the moray. It's a tiny little siphon. And then here on the mola, it's just a tiny little flap right next to the fin. So really different than what you're used to picturing on something like a typical fish where you've got like a big gill opening, usually right around the operculum. That's why it's called the opercular opening traditionally, because it's right near that operculum, and it's usually quite large. But in a lot of fishes, it's actually reduced to a really small opening. And I wanted to introduce you to the anatomy of the gill chamber. So we've got the buccal pump. Hopefully you're familiar with the jaws. We've got the buccal system is relatively familiar to people, but the opercular system, the gill chamber, might not be so familiar. So we've got the opercular bones, and that if you ever watch a fish breathe, you can kind of see that flapping back and forth. And those are the opercular bones that you're seeing. The opercular, subopercular, and interopercular. And underneath those bones, or ventral to the bones, are these structures that expand and contract. And they're my favorite structures. These are the branchiostical rays. So I really mentioned my students call me Dr. Branchiostical because I talk about these things all the time. I think they're the most underappreciated structures in fish biology. And what they do is quite important. So they sit underneath the operculum and they expand and contract. And they're different sizes in different fishes. So most of the time when you're looking at fishes, you're going to see that operculum flapping back and forth. But next time you watch a fish breathe, look for these branchiosticles, and I'll put them out in a bunch of different ways throughout my talk. But they actually do quite a bit of pumping. So that expansion and contraction, it helps to maintain that positive and negative change in pressure during ventilation. So that's basically the anatomy of the gill chamber. All of those structures together form that gill chamber pump we've been talking about. But to understand restricted gill openings, I wanted to back up and understand the anatomy first, and I wanted to understand it specifically in an evolutionary context. So we see a bunch of fishes that have restricted gill openings. Where are they distributed on the tree of fishes? And by the way, we're talking about ray-finned fishes here, and these guys are they are about 30,000 species, and they make up maybe about half of the approximately 60,000 species of vertebrate diversity. So these guys are all of the fishes except for sharks and things like lungfishes and coelacanths. But there's a huge amount of diversity. So I think the number is like 33,000 species now. And so I wanted to see across the tree of fishes how many times restricted gill openings have evolved, and whether or not it's correlated with anything specific, whether or not we can tie it to any aspects of morphology or ecology that might tell us more about what those gill openings are doing. So I talk a lot about the membrane that surrounds the branchiosticles. So these branchiostical rays, so that's what these are here. You can see this really nicely on the spoven, and it's covered in this membrane of connective tissue, and then there are muscles between the rays. But this membrane can be have 4 different configurations. So it can either be the membranes can be sort of separated on the right and the left of the head. So these 2 membranes are totally separate on the right and left, and they're also separate from the body wall. So that'll become a little bit more clear in a second. But so that's called separate from the membranes are separate from one another, and this is membranes are united to one another. So you can see they make this one continuous membrane, which is really interesting. And then I'll show you the other 2 morphologies that I was interested in, which that membrane can sometimes become attached to the body wall. So maybe hopefully this makes it a little bit more clear. So now we've got the 2 membranes are separate from one another, but they're also stuck to the side of the body. I call that it's called the isthmus, and that's basically the bottom side of the fish. And then if that attachment gets really, really broad, you wind up with a restricted gill opening, and that's basically how you get restricted gill openings. So so the way I approached this question was I did a big survey to look at how many times this has evolved. So I studied 433 families, recorded their morphology, and I used this color scheme. So I use this color scheme throughout for separate united. The membranes are just joined to the isthmus, and then membranes joined to the isthmus, but restricted gill openings in red. So the main color I want you to focus on is the red, because that's the restricted gill opening fishes. So to put together the tree, I did an ancestral state reconstruction. So I looked at where in each node of my tree that I was studying, where you winded up with an ancestor that had restricted gill veins. So I'll show you what that looks like in a second. But to construct a tree, I used a previously gathered data set by my collaborator Tom here, and on the phylogeny there were 284 families represented, and to construct the tree he had used 9 nuclear markers. And so I took a subset. So his data set's actually quite a bit bigger, but I took one representative from each family. I ran it through Beast. I came up with the maximum clade credibility tree, and I used this function in the 5 tools package. This is problem. This is why there's a DVD player on this cable. This cable is going. Actually, I think we might switch over to. Oh,
There you go.
all right. Hopefully that doesn't happen again. Sorry, guys. This is the method that I use for ancestral state reconstruction. It's a little bit complicated, but I'm happy to answer any questions about that. So this is what the ancestral state reconstruction looks like. So you see these little dots of red. So these pie charts represent what the probability of the ancestor being the particular condition of branchial membrane morphology. So when you see a lot of red, that means the ancestor was almost certainly an ancestor with restricted gill openings. So anytime you see those red pie charts, that means that it's an example of convergent evolution of restricted gill openings. And you can see it crops up all over. There's only really a few parts of the tree where there aren't fishes with restricted gill openings. And we pinpointed, using this technique, we pinpointed approximately 11 instances of convergent evolution of restricted gill openings. And I want to draw your attention to one particular part of the tree, which is this cool clade that I happen to really have a fondness for, which are the tetradontiforms and the lophiiforms, which are the lophiiforms are the anglerfishes, and there's the frogfish and the deep sea angler. And then the tetradontiforms are things like triggerfishes and pufferfishes and the ocean sunfish. And the cool thing is that this recovery of this clade as a whole big group where these two lophiiforms and tetradontiforms are sister to one another is actually a fairly new arrangement. So people are just starting to explore really what are some morphological features that these two groups share. And so it was really exciting to be able to document this in my paper as not only a cool clade, but also an instance of convergent evolution of restricted gill openings. So you can also see the pie charts a little bit more clearly on the slide. So you can see, so we have a bunch of blue, and then we see this big transition to restricted gill openings, which I think is really interesting. You see that all over the ray-finned fish tree. So now that I know that it's evolved a bunch of times, I was curious as to what sort of morphologies these fish had. And so every time I measure fish, and now these are back to being 433 families, that was 284 on the phylogeny, but I was able to put all of my taxa into this principal component analysis, and I did that by taking a bunch of measurements of every fish that I came across in my survey. I measured things like jaw size, snout size. I obviously did some things to quantify the size of the gill openings, but I also measured the length of the operculum, measured body length, I measured fin length, and I put all of these things into a principal component analysis, which basically takes all of your variables and sort of summarizes how they relate to one another. And what you can do then is create what's called a morphospace, where you now have plotted sort of a distribution of how all of your anatomical characters are related to one another. And I just sort of put, just to orient you, these guys over here are the really highly elongate fishes. Those guys there are fishes with like abnormally gigantic mouths. And these guys are, those are like the anglerfishes. And the tiny mouths, long snouts are fishes like tube snouts and pipefishes and seahorses and things. And the elongate fishes are obviously things like gills. And one of the things that jumped out at me when I plotted this was that there was really no one area that fishes with a restricted gill opening occupied. Instead, and I have circled each of circled all of the points for each morphology of the membrane. And what I found is that fishes with restricted gill openings actually occupy a huge morphospace, much larger than fishes with other morphologies, even though there are only 80 families out of the 433 that I studied that actually have this morphology. Those fishes that had restricted gill openings tended to be on the outsides of that morphospace, which I found to be really interesting because that says to me that maybe restricted gill openings allow you to modify your cranial structure in different ways so that you end up with these really wacky morphologies. And I'll talk about that in just a second, but I wanted to make one more note while I have this graph up, that the anglerfishes were up in this corner and the tetrodontiforms, the triggerfishes and pufferfishes, were way down here. And so even though these guys are really closely together on the tree, they actually ended up on sort of opposite ends of my morphospace. So I was pretty confident that I was capturing a lot of the morphological variation. So anglerfishes have really big mouths. Tetrodontiforms have really tiny mouths and long snouts. So, so I just wanted to then look at ecological data, and I did a little bit more of an analysis. But what I generally found is that you can find fishes with tiny gill openings pretty much anywhere. You can find them in all sorts of salinities, all sorts of regions across the globe. You can find them in the deep sea. You can find them in super shallow water. The only place where it's rare to find them is the open ocean, and the one exception to that is the ocean sunfish, the mola mola. The typical form for the open ocean is a fish that's swimming up in the water column, is doing a lot of ram ventilation, high energy fish, and mola doesn't really fit that that mold really. If you remember the video, that was the guy on the bottom. And so I think probably if you have a restricted gill opening, living in the open ocean is probably not the best idea. But that was pretty much the only trend that I saw in terms of my study of the ecology of fishes with this morphology. And then I was interested, as a functional morphologist, I'm interested in why fishes would have these gill openings. Unfortunately, I don't have a conclusive, satisfactory answer. But what I think is that these gill openings mean different things for different fishes. So some fishes have put their gill opening behind their pectoral fin. Some have put it on the top of their head, and that has allowed some extreme cranial morphologies to evolve. So being able to move your gill opening wherever you want has, or I shouldn't say that so assertively. It's potentially could allow some variation in how you set up your skull by not being constrained by where you put your gill opening. Also, once you have your gill opening, once you have that really broad attachment to the isthmus that we've been talking about, you have lots of space for things like sucker discs and big ventral keels. And there are a couple fish that have that morphology that have restricted gill openings. And also, people always point out frogfish actually use their gill openings to jet around, so they can actually push a lot of water out of that. And because it's such a small little hole, they can actually get quite a big jet going. So gill opening jetting, and potentially there are some, like the mormyrids are electric fishes, and potentially having a tiny gill opening might allow them to have less disruption to their electric field as they're communicating. And also there are a lot of the most extreme rapid suction feeders have tiny gill openings. And fishes that have, some fishes with those protrusible pharyngeal jaws have isolated, so like the moray eels that can actually stick their pharyngeal jaws out of their mouths have isolated their pharyngeal jaws from the rest of the gill chamber, forming the gill chamber into this weird sac. And I think that restricted gill openings are necessary for that. There's some cool data coming out on that from the Rita Matus lab. And also potentially it could influence, having a tiny gill opening could potentially influence the basics of flow in terms of how these fish are generating pressures. So because I came to this conclusion that restricted gill openings mean different things for different fish, I'm probably not going to be able to answer that question. So I decided to focus on what restricted gill openings mean for one specific fish. I'm just going to take a drink. I'm going to let you appreciate, actually, no, I have a video for you. So really quick, these guys are really weird looking. They're called monkfish because they needed a fancy name for people who actually want to eat them. I found out you can get monkfish livers quite commonly in restaurants. But they're these really sort of low metabolism fish. They have very, like, the reason they're so savory is because they really don't move around a lot. So they're very little red muscle. And they take water in through this mouth, and then it passes through this giant gill opening, which we'll talk quite a bit about. And then it goes underneath the fin and comes out this opening, which is way behind the fin, which which is really, really unusual for a fish. But these guys are really flat. They just sit on the bottom their whole lives. And when they exhale, it looks like this. It's automatic. There's this little tiny hole that sort of pops open. I'm just going to do this one more time. It's behind the fin, really unusual. I call them armpit breathers sometimes because, like, basically breathing out their armpit. But it's really quite spectacular. And so I brought a bunch of these guys to the beautiful Shoals Marine Lab, and I mean, you can have the worst camera and take pictures out of Shoals and they'll all come out like this. It's really an awesome place to hang out. And I was really fortunate to be able to go out there, and the staff were wonderful, helping me with keep my fish and take some data from them. And also having this beautiful view to look out at every day was also pretty wonderful. And so I brought these fish out to the lab and studied them. I basically took just a ton of video to try to quantify and examine what they were doing in terms of their ventilation. And one thing that's been noted about them, and one thing that I found right off the bat, is that when you're watching them, the video is playing, they're just not doing very much. So when you're looking at them, this is what they look like. Basically, you really can't see much of anything. They're just sort of hanging out. And for a long time, people thought that all they did was just exhale every once in a while, and otherwise they were just sitting there, pretty much doing nothing. But when I sped my videos up, so this is 20 times the normal speed, this is what I saw. So now these are cycles that are taking about a minute to complete, and I've sped them up 20 times, and you can see that the fish is pretty much moving throughout the entire cycle. I'm going to play that one more time. So you can kind of watch the margins of the fish and see it very slowly moving upwards, expanding, and then it'll exhale about once a minute. So I was curious as to what that movement actually is. What is the fish actually doing during that period that it's doing just so incredibly slowly? So what I did was I took my videos of my fish breathing, and I tracked a bunch of points. And I'll talk about what those points are in a second, but basically this process is called digitizing, when you pick points on the body and you track them across a time period. And so I took those and tracked them for different ventilatory cycles. And what I found is that there is quite a bit of movement throughout the cycle. It is just really slowly. And this is, I should point out that this is a quicker ventilation cycle, and I'll talk about that in a minute. Actually, I'll just introduce it really quickly. So these fish were breathing about once every minute to sometimes 2 minutes, and the longest cycle I ever observed was 3.5 minutes, which is pretty extreme. So that fish is basically filling up its gill chamber the entire time. This blue here represents the phase where it's basically filling just the gill chamber, so it's really, really slowly expanding its gill chamber. And you can see that phase takes up the majority of the cycle, and then we see this little blip where it's exhaling at the very end. But whenever the fish was disturbed, so they build these little sand recesses, and that's when they're really happy. They just sit there, they dig out their little sand recesses, and then they start doing their cryptic ventilation. And I call it cryptic ventilation because it really does reduce the movement of the fish, and it makes it really hard to see the fish, and it makes it really hard to observe any sort of movement that the fish is making. But if a fish is disturbed from that recess, if you really aggravate it and it's forced to swim around, it's not super happy about it. And so it'll breathe like once every 20 to 30 seconds, which is actually quite rapid for these guys. I would talk about how stressed out my fish were because they were breathing once every 30 seconds, and people were looking at me like I was insane. So this is actually a faster cycle because I had to disturb the fish from its sand recess to be able to do this digitizing. But throughout the entire cycle, and basically the most dramatic thing was this head lifting. So this is tracking just the movements of the eye. And so the fish starts by doing, these graphs are related to one another. And you can see a bunch of movement. The blue is the most important one. Red is the jaw, just so that you know that basically throughout the blue phase, which is when the fish is inhaling, the jaw really isn't doing anything. It's staying stable. And that is jaw position relative to the eye. So we've adjusted for head position when we've digitized on this top graph. And the blue is the operculum. So you see this really big expansion of the operculum at the beginning of the cycle, so they flare up their operculum pretty slowly, but they do flare it quite a bit. And then they switch over to this head lifting, and unfortunately I couldn't digitize the branchioscles. But what this basically is the branchioscles are expanding, and the fish is lifting its head to make room for those rays as they are expanding. I'm actually going to show you really quickly what those rays look like. So these are the branchioscles here, and they actually go underneath the fin, despite this picture. And so they have huge branchioscal rays, and they just very, very slowly expand them. And I also this is kind of a fun photo. When they exhale, they really shoot a jet. So they take in this really large volume of water, and then they're able to shoot out a very large jet to get rid of that water quickly so that they can go back to being cryptic. So they have these huge branchioscles to contribute to this expansion. And I wanted to show you kind of what they look like on the fish itself. So this is Willie's arm inside the gill chamber of a 66-pound monkfish. And so they have a very, very large gill chamber. So this can hold a very large volume of water. And so these are the branchioscal rays that you can see, these indents here. And you can see they're very long, relatively robust rays that these fish have. Oh, and by the way, I should point out, these are their pelvic fins. So it looks like the length of branchioscal is at least twice the length of the pelvic fins. Really weird. These guys are super cool fish. And they also, like most lophiiforms, they take their pelvic fins and they move them and use them to walk along the bottom. Mostly they like to sit around. So the other thing that I did with these fish is I did some anatomical work on the gill opening. So this is that 66-pound monkfish that we dissected. And here's the gill opening here, behind the fin. So we know it's sitting behind the fin. And what we've done now is we've skinned the side of the body. And you can see underneath this is all the meat that you would want to eat if you're actually trying to eat these guys. But we also found this, actually I should use this from here, but I don't know. I like gesturing inside, so let's try. So there's this really thin band of muscle. It's a little bit hard to see, but it's actually sitting on top of all these trunk muscles here. And it runs from the gill opening all the way up the body. And I just want to show you that it actually is a thin sheet that's embedded in the connective tissue on top of the body. So it's not a part of this really robust trunk musculature that everybody's used to eating. It's actually this really thin band of muscle, and it forms, these are the dorsal fin rays, this is the dorsal surface of the body. And it actually forms this muscular sling that goes over the top of the body. So you can imagine it going all the way across here. And what these muscles are, that's bad. I don't know if there's a way to improve that contrast. But this is a photo of muscle fibers. Can somebody turn on the lights? I think that might help. Maybe the lights are, oh. That's unfortunate. Anyway, thanks, Kelly. I think I'm just going to go with this for now. So these muscles are coming from so these are branchioscal rays here, and the muscles are actually muscles from between the branchioscal rays, and they come and they normally just span the width of the branchioscles between them, and that's what causes that big contraction. But these they're muscles at the very, very tips of the branchioscles actually expand out, and those muscles, we've traced them from between the branchioscles down here, and then they go out and they expand to this really large gill chamber musculature. So this is some really cool morphology, and what gets us excited about it is that it's one of the first fish where we've documented some really weird muscular control of the gill opening that's never been seen before. And we think that this musculature contracts because it's muscles that are involved with contraction during the exhalation phase. We think that it's involved with contracting not just the gill chamber, but pulling that gill chamber, pulling the gill opening open so that it forms that nice little siphon when the fish is exhaling. The other really exciting thing about it is that tetrad nonconforms also they don't have quite this dramatic expansion of this muscle, but this muscle the muscle that comes from between the branchiostral rays does expand into the gill chamber of the triggerfishes and pufferfishes, and it actually can get quite big. And so potentially this could be a morphological figure that unites those two groups, which is something that we're really excited about. So for the last system that I ended up working on, I decided that monkfish were really so weird. And I probably needed to switch to something where a little bit more, these fish are looking a little bit more like typical fishes here. These are the cottoids, and they are the sculpins and the poachers, and the like parrots and lumpsuckers, and also sandfishes here. And the reason that I chose this system was because they are really diverse, and they also have a whole bunch of different morphology of their gill chambers. So I decided to go back to my sort of original questions about not just how really weird fish breathe, but how normal fish breathe and how it varies among a clade of relatively closely related species. So again, I got to go to a beautiful place. This is one of the beautiful pictures you can take at Friday Harbor Labs, and this is also where a lot of species of sculpins can be found. We have about within the cottoids, we have I think about 40 species out that you can get either intertidling or doing subtidal trawls or speech scenes. And so this was an awesome place to be able to explore some of this diversity. Sorry. And so these are the 4 species that I ended up working on for the first part of my study. And these are CT scans done here at Cornell, and hopefully you can get a quick sense that the gill chambers have a little bit of different morphology in each fish. And what I want to draw your attention to is differences in the morphology related to the size of the operculum. So which fish have really big opercular bones relative to the size of the branchiostral rays. So this guy has relatively big operculum and branchiostrals. This guy has a pretty small operculum up here and huge branchiostrals. And we'll go into that a little bit more in this next figure. So what I generally found is it wasn't a true continuum, but within these 4 species I was able to get in the lab, these guys have really huge opercular bones and tiny, tiny branchiostral rays, and spiny-headed sculpin has really small opercular bones and large branchiostral rays. So even within these 4 species, that they all look pretty similar, they're all really closely related. They had some morphological differences, and so what I wanted to do is try to understand how pressures varied when they were ventilated. So now I switched to doing something a little bit more related to form and function, which is to actually measure pressures in the head rather than just looking at what the fish was doing in terms of moving its skeletal elements. And so these are pressure recordings. I just wanted to sort of orient you to what I actually did. So this is pressures in the mouth. So I planted transducers in the mouth and the gill chamber. And then I subtracted the opercular pressures from the mouth chamber pressures and got a differential pressure, which really gives you a picture of how the pressures are different relative to one another at different parts of the cycle. So which chamber is sort of contributing more pressure to a particular phase. And so just to really get into this now, I've shown you this graph and or this diagram, and now I'm kind of going to really dissect what these fish are doing. So I've got pressure transducers in the mouth and the gill chamber, and what I'm interested in is what these different pressures are doing at different times. And so just to go over this again, this phase 1, I refer to this a lot, and this was that really long phase in the fish. And what they're doing is this is when they've already started to expand their gill chamber, and then they really start to expand their gill chamber, and then so this is phase 1, and then phase 2 is when they're basically switching over. So now they start to compress their mouth while they're still expanding their gill chamber, and then in phase 3 they start to they're compressing both of their chambers. So they've already started compressing their mouth, and they start to compress their gill chamber, and then phase 4 is when they switch back to the suction phase. And I'm really focused on phase 1 and phase 2 because that's when I know for sure that both chambers are generating either positive or negative pressure. So these phases get a little bit difficult to understand in terms of comparing pressures because you're not really totally sure how to interpret these differential pressure profiles. And again, when I say differential pressure profiles, that's opercular pressure minus buccal pressure. And that's basically what I'm going to be working with when I look at my data. And so these are differential pressure profiles of fishes that were studied by G.M. Hughes in the 60s. And he set up this dichotomy of fishes that live up in the water column, so pelagic fishes swimming around. They do one thing, and then benthic fishes do another thing. And benthic fishes are fishes that sit on the bottom. And so he basically came to the conclusion that pelagic fishes have this big peak during phase 3, which again is that positive pressure phase. And benthic fishes have a big peak during phase 1, which is the negative pressure phase. And what he concluded is that pelagic fishes mostly use their mouth to pump, and benthic fishes mostly use their gill chambers to pump. Even though they're doing all of the normal ventilatory movements, one pump is dominant in each different type of ventilation. What I found when I looked at the differential pressures of my fishes is that really it varied way more than I was expecting. So Leptocotis armatus was the fish with really tiny opercular bones, and he was basically just, they were buccal pumping the entire time. They were really, really powerful buccal pumpers. And these two species here were really powerful opercular pumpers, so they were really pumping heavily with their gill chambers. And Dissertation in particular had this really, really long, you can see that the time has been adjusted so that you can see this is actually 4 seconds. They had a relatively long phase 1 relative to other fishes, and they also had a really big negative pressure being generated by that expansion of the gill chamber. So throughout these 4 species, there was actually quite a bit of variation in whether or not they had a pelagic profile or a benthic profile. Even though all of these fish are benthic, they all like to sit on the bottom. They would all be benthic by anybody's definition. So that was really interesting. And then we wanted to understand how these, the morphology related to back to these, whether or not they had a buccal or whether or not they had a buccal pump or a dominant gill chamber pump. And we ran into one problem that's really common in biology, is that we're looking at 4 species. They're not all independent data points because you have relatedness among the taxa. So these 2 fishes are more closely related to one another than they are to Hemilepidotus, and then these 3 species are more closely related to one another than they are to Leptocotis. So we did a phylogenetic correction, and once we did that, we were able to do a least squares model. So like more or less, we were able to plot the points against one another and assess the significance of the relationship between them. And so we did this with a bunch of different anatomical variables, things like jaw size. We actually tried to quantify the volume of the different chambers relative to one another. So we made molds of the chambers, and pretty much nothing correlated with what we're calling the pump ratio, which is I'm happy to explain that, but it's basically this metric that we came up with to describe whether or not you had a pelagic or a benthic pressure profile. And the only thing that really correlated was the size of the branchiostral membrane, which is basically just the size of the branchiostral apparatus relative to the size of the operculum. So if you had a big operculum and a small branchiostral apparatus, you were down here. So this is Leptocotis here, really strong buccal pumper. And then if you had large branchiostules and a small operculum, you were up there like Hemilepidotus and Dissertation. So there was a really, really strong signal, even though we only had essentially 4 data points. These are species averages. We still had a significant regression. And so what we've been talking about is how do we take that information and think about how we can modify our understanding of gill ventilation. And what we've decided is that because of the relative size of these structures is so important, especially the relative size of the branchiostral rays, we decided that you can get a lot more information by modeling this as a 3-pump system rather than a 2-pump system, where the opercular bones and branchiostules, they're not doing exactly the same thing. They're working together, but if you remember what we saw in Lophius, this was an extreme example. The operculum was going at one time, branchiosticles were doing something a little bit different. And so they are very related to one another, and they are doing essentially the same thing, but they're working together more than they are working as one unified pump. And so just by modifying the model in that way, we can get a lot more. We can develop our understanding of gill ventilation a lot more clearly, especially how it varies among different fishes. I don't want to dwell on this for too long, but we have been doing some physical modeling of this. This is version 2.0, 1.0. We have some data from this, but I'm working on 2.0, and it's coming along, but it didn't make it into my dissertation. But we're trying to verify some of the adjustments we've been making to this model by actually physically making models and seeing what they do at Atlantic pressure transducers and doing some experiments. Another thing that we'd like to do, so this is for my postdoc. I have a PRFB, a postdoc research fellowship in biology, to study the microanatomy of gills. And so you can just kind of get a sense. The top are from fish that migrate quite a bit in the water column, and the bottom gills are actually from a deep sea eel. And there's quite a bit of variation in the morphology of these structures. So I've been really focused on the pumps for my dissertation, but now I'm trying to make the switch to thinking about what's happening at sort of this microscopic level. I've been working with the CT lab here to get some preliminary data, and these are actually this is actually rendered at 2 microns. We can actually go have a little bit more resolution here. So these are these super tiny structures, and you can see these scale bars. That's 20 microns long. These structures are about 5 microns long, and we can actually make CT models. And here's a nice, pretty CT volume rendering. And here's a surface model that we can actually make. So for the first time, we can take these models and we can actually put them into computational programs and actually understand. I've been talking basically about the gills as just this like curtain of resistance between the 2 chambers, but really it's a lot more complicated. So that's something that I'm interested in getting into. So I do have a 4th chapter. I'm 46 minutes in. Go for it. All right, you guys are still with me. Okay, it's quick, and one of the things that's really fascinated me about the system is the fact that every single fish needs to breathe, but every single fish needs to do a lot of other things with their head. And so I switched gears with my 4th chapter to start thinking about how the ventilatory system might evolve, not just as it functions just for ventilation, but also as it's sort of changing to do other things with the head. So we've seen now that changing your gill opening allows you to do other weird things with your cranial morphology potentially. But how does the gill opening relate to? How does the gill chamber relate to other aspects of cranial anatomy? So one of the major things that a lot of fishes do, and all of my sculpins do, so this is going to be another sculpin story, they all suction feed. So they all take their mouth structures and they expand them very, very quickly. And that draws a lot of water into the chamber by just really rapidly creating a bunch of negative pressure. So we know now that the mouth is really heavily involved with ventilation, but it's also under a lot of selective pressure to do this suction feeding really efficiently. So how does the gill chamber sort of play into that? One of the things that I want to point out is that the opercle, we've been talking about it just in the context of gill ventilation, but it's a really important structure in the context of suction feeding. And the reason that is because you might notice that this interopercle bone is actually linked in with the lower jaw. So there's a connection between this interopercular bone, which is a part of the opercular series. It's actually connected to the lower jaw. And there are muscles on the operculum that contract, and when they do, they rotate the entire opercular system, and it contributes. It doesn't flare out the mouth entirely, but it makes a major contribution to expanding the mouth during suction feeding. So this is a pretty well-known thing that the opercle is a really important structure in not just ventilation, but also suction feeding. So what does that mean for the evolution of all these structures? How do you have, they're all evolving obviously in the same animal, and so how do you begin to understand how different structures that are doing different things are evolving to make one head really good at what it does? So I have two different hypotheses, and I'd like you to think about them as separate for now, because joining them is something that makes my brain hurt. But suction feeding is, to understand how a suction feeding system might evolve, you think about an animal that's evolving to be really good at suction feeding. So the sizes of the upper jaw, lower jaw, and opercle should all be evolving at the same rate and together at the same size. So like as the fish gets a bigger mouth, all of those structures should become bigger. And as the fish evolves, there are some awesome suction feeders that have like really, really tiny mouths, and that they're sort of like they like to feed out of a straw. They can do it really quickly. Everything gets smaller all at once. So these shifts should happen all at the same time. And in gill ventilation, we expect the size of the opercular and branchiospitals to be correlated with one another. So we know a little bit now that the size of the opercle relative to the branchiospitals can change a lot, but maybe like if one gets smaller, then it's bigger. If one gets bigger, then it's smaller. Or maybe one gets bigger, they both get bigger. So we expect there to be correlation between these two structures. And so these are sort of two different hypotheses that we can actually test by using comparative methods. And so this is, again, just reorienting you to the group that I'm working on. Really awesome, really diverse. And we have this, this molecular data set. I used an existing data set from Matthew Nope that had 99 species of Catoideae, and I also added some species, especially outgroup species. And I also added another set of genes just to make the tree a little bit more robust and add a little bit more data. And then I used Bayesian reconstruction. I used Mr. Babes and created a maximum clay credibility tree from those runs. And the tree actually looked pretty nice. So the things to look for when you're assessing whether or not your tree is a good tree is whether or not all the nodes are resolved, and whether or not at each node you were able to recover a high probability of that node being a real thing. And this is the prune tree. So then I pruned it down to 24 taxa, because those are the taxa that I had morphological data from. And I took a bunch of measurements on these fishes, but I'm just going to go over a few. And these are just related to the hypotheses that I was trying to test. And so this is some of the things I measured were size of the upper jaw, size of the lower jaw, the mouth opening diameter, the distance between the two jaw joints, and the length of the opercle, and also the average branchiocipal length. And I took that I didn't really explain it all that well, but the phylogenetically corrected generalized least squares models that I was using for my first data set, I applied the same sort of techniques here. So where I corrected for phylogeny, I corrected for all those relationships. And these are p-values, so these are significant studies. And actually what I found, and I think this is probably, I have a couple different theories on why all of the values that I found were significant, but the values of correlation between the jaws, the two jaws, the mouth opening diameter, and the opercular length, they were all highly correlated with one another. So those are all these dark boxes here. High degree of correlation between the jaws and the operculum. But when you looked at branchiocipal length, it didn't really correlate with anything. The values were sort of significant, but they weren't, highly significant. And the first thing I did point out already that opercular length is basically correlated with all these three dimensions of the jaw that we've recorded. And so that we think that that means that the suction feeding structures are highly integrated. So that means there's a high degree of correlation. So that's something that's really cool. And it's pretty well known that we expected these structures to be highly correlated with one another. What we didn't expect is that branchiocipal length is really not correlated with anything. We might even expect it to be correlated with the lower jaw because the jaws are involved with ventilation. But it actually wasn't really correlated with anything. And so we're calling this an example of evolutionary modularity. So basically, these branchiocipals are not really tracking with anything. They're just kind of doing what they're doing. So while the fish is sort of evolving to suction feed in different ways, the branchiocipals might just be like compensating. So like if the fish needs to do something with its feeding system. Branchiopterals might just change in size in ways that supports specifically ventilation. So if you're interested in sources of variation in gill ventilation biomechanics, that might be why we're seeing branchiopterals come up as these important structures over and over again, because they seem to be the one thing that doesn't really track with any other aspects of the head that are involved with feeding. So potentially they're sort of a separate module. That's where this term evolutionary modularity comes from. And we'd like to do a little bit more rigorous quantification by taking a bunch of measurements off our CT scans and doing this again, just to make sure that we've really assessed this, because potentially this could be a really interesting way to understand the evolution of cranial anatomy within the fish skull. And, with that, here's my final slide. And I have a ton of people to thank. So all of this took a ton of work, and I couldn't have done any of it without a whole bunch of people supporting me. And oh, actually, you know what? I left my old. Okay, so this is my new thing slide. I did a little bit of restructuring just to make things a little bit more easier for you guys to understand. So I would like to thank my committee for being super supportive, and I'd also like to thank John Friel, who was on my committee, but he recently took a position at the Natural History Museum of Alabama, and so he's unable to be here. But I also have a ton of other people that have supported me in a mentor role. Frank Fisher, we love it, definitely supported me in a teaching role. Tom Neer taught me a whole bunch of stuff about molecular data, and Beth Brainerd in my committee have definitely provided a lot of intellectual guidance. And I also want to give a special thank you to my undergraduate collaborators. So the undergrads are the three with their date of their graduation. And I call them collaborators because we've sat down and I've written with all three of them. So Will has now been published in the Journal of Experimental Biology. Catherine wrote an awesome paper for the phylogenetics class, and we're working on a manuscript for stuff she's doing in the summer. And Noah Braxton, we just submitted a paper to the Journal of Experimental Zoology on jumping fishes, which we also worked on with Alice Gibb. And Matthew Nope and Laura Ferry, we've also done some writing and presenting, especially with the sculpin stuff. So Matthew Nope has provided a whole bunch of data for understanding sculpin phylogenies. And I also couldn't have done any of this without the awesome staff and the people at the different marine labs, so Shoals Marine Lab and Friday Harbor Labs. It's really been awesome to be able to work at these places, and a whole bunch of people have helped me out at various times working there. And also, I've gotten a bunch of statistical help. I took Elizabeth Murray's class in phylogenetics, which was huge in helping me get through these big data sets where I had a whole bunch of data that I wanted to look at in a phylogenetic context. And also just I've gotten statistical help from a bunch of people over the years. And I also would love to thank all of my funding sources. I've gotten a ton of really great support. The doctoral dissertation improvement grant basically sort of skyrocketed my ability to go out to Friday Harbor and do a bunch of cool stuff. So I'm really grateful for that. And of course, Shoals and Friday Harbor, couldn't have done it without them. And here are just the glamour shots of my various mentors. And yeah, sorry that really didn't show up this year today, but it did exist at one point. Here's the documentation of that. And here's the three students I've been talking about: Will, Noah, and Catherine, are the students I've written papers with, but also these other three columns are from the fish course at Friday Harbor that I TA'd, and I've had just a bunch of really great undergrads, students that I've worked with in classes and on campus at Shoals at Friday Harbor. It's really been awesome to see these guys. They keep things fun, but they also are really serious scientists and really cool people. And also my family. So my family's here today, and my wonderful husband has been very supportive in the collections, helping me count brachial stories and stuff like that. Really just been there through the whole process. And with that, I'll take your questions.
So we'll take some general questions, or Stacy will take some general questions, and then the committee will go off and talk to you in a couple hours. I'll work.
So I was curious about your reconstruction of your 11 reduced gill openings.
seemed to me like the ancestral state was actually blue, which I presume separate two little straps on each side of the head. And I wondered whether you had actually looked at the transitions, because I mean, would you expect some sort of ordered evolution of those 4 states?
That's a really great question. The only hypothesis of order that I actually have, and this is like my first question, so it's really cool. The only hypothesis that I actually have from an anatomical perspective is this transition from that join to the isthmus to restricted gill openings, because that transition makes a lot of sense to me. So first you get that attachment to the isthmus, and then you can end up with a really tiny gill opening. In terms of whether or not the ancestral state is actually united versus separate, this is the base of my tree, and this is pretty inconclusive. And there's actually, even within, let's see if there's a group. So there, within I showed that the gar and the bowfin, and they are sister clades, and they're one of the most ancestral groups within fishes, and they still have this. And one has united and one has separate. And then if you look at the outgroup to all the Actinopterygii, you see the things like rockfishes. They all have separate membrane.
So is that the base of the tree? Because I saw the blue in the tree.
Yeah, let's go back to the big tree here. I'm just gonna.
That's.
Yes, there's a ton of blue, especially when you get down. These are like this hodgepodge group that's been called just like the Perciformes. It used to be wildly paraphyletic, but now we're starting to parse it apart. But there's a ton of blue, so it's definitely the united membranes are the most common. But I think it might be separate at the base of the tree. I can also sort of think about what other like Sarcopterygians are doing.
So you've reconstructed those states already, but the crazy thing that I see is that there's a lot of transitions from blue directly to red, right?
Yeah.
So I guess what I was asking is there a correlation between any pair or groups of those transitions there on your tree? You just said that maybe from yellow to red would be the one. But you don't find that? You don't find that as a pattern? The transition to from yellow to red, you said anatomically would be more likely.
Exactly, but I don't see it a lot. So I see it here and I see it here. But yeah, I would love to talk about some sort of, is there some way to quantify these transitions? That would be really useful. Because right now I'm just treating them as all the same. I'm not ordering them in any way. So that might actually help me, like ordering them might actually help me figure out which one is the ancestral one if I can figure out how often.
Yeah, because we quantify it under different models of expected ordering, and then see which one fits better.
Yeah, definitely. Cool. Yeah, let's check on that. Nick?
Yeah, also related to this stuff, you were mentioning how, this is already cool, how the restricted gill opening might facilitate diversification into other functional niches, freeing up space in the face and stuff like that. Is there any relationship or association between different gill openings and species richness across these fish families? Does this also relate to diversification patterns of fishes?
Yeah, that would be a great next step, and I would really hesitate to say that restricted gill openings causes diversification. Like, I'm trying to come up with a way to describe this sort of like extreme shifts in cranial anatomy that may or may not relate to diversification. So I don't have a sense I don't have numbers off the top of my head, but I don't get the sense that this big clade of restricted gill opening fishes is any more diverse than some of these other clades. But that would definitely be something that would be awesome to quantify. I think the only really diverse group that has tiny gill openings are the snailfishes, the Liparids, which is something that's been coming up a lot in literature on diversification. They're wildly diversifying. But there doesn't seem to be a trend of restricted gill openings causing speciation that I point out. Yeah. Awesome thing to think about.
Sure. Just natural history. That fish that it had cryptic ventilation on that fish. What about, is there any convergence when you look at other ecomorphotypes, that fish that sit on the bottom like that, and do they have this similar, I mean, is this something new or is this something?
This is the most extreme, so I don't think that there are any fishes that breathe quite this slowly. I think there are some catfishes that have been documented to breathe pretty slowly, but I think it's mostly because they lower their metabolism. These guys, they don't bury. They sort of throw sand on their backs, but they don't actually like bury themselves. So the only thing the only thing that's ever come close to being this slow are these that I've found are actually these Dasycotis. So let me just. So these guys, this cycle took about 5 seconds, but I've seen these guys go about 20 seconds before completing a cycle, and they seem to do the same thing. You can see this is starting to really look like a low phase ventilation cycle here, where they have a really, really long phase 1. And this is the only place I've really looked for it, but I wouldn't be surprised if other fishes that are extreme bottom dwellers have a really slow ventilation. A lot of them have really big branchiospitals, but it's definitely not the rule. Flatfishes are sort of laying on their side, and their heads are so weird that they kind of can't have big branchiospitals. But I'm sure there are more examples that I can go out and look for. I'd love that. It would be really cool if it was an actual trend in terms of this extreme benthic morphology.
Well, Nick, you have another.
I do have another. For your last chapter, all of those correlations you're looking at among your different feeding morphology traits, are how much variation is there in body size across these things in zoometry?
So what I did, and I didn't describe it here, but I did a phylogenetically corrected method of doing size corrections. And this was, I don't know if you've, so Liam Ravel's method of correcting for size. So in theory, these should be all corrected for any issues related to the size of the different fishes.
Well, Stacy, I have to tell you, it was wonderful to hear this, and it's amazing to me that you got 4 big projects done in such a short period of time. And I want to thank you for a terrific seminar. And the committee will meet, I guess, well, is that right? Everybody got that on the calendars up in the cold room? Well, right now, we'll thank you very much for a terrific talk. Yeah.